This is a B3 video from the topic infection response, looking at some triple content, and we're looking at monoclonal antibodies. We're going to look at producing monoclonal antibodies and then uses of monoclonal antibodies. So why do we even need monoclonal antibodies? Okay, white blood cells can only make one type of antibody. So if you imagine all your white blood cells, some of them can make the antibody for one disease, some of them can make the antibody for another disease. Okay, so they can only make one type of antibody. They cannot make other antibodies. So we've got these little clusters of white blood cells that can all make their own particular antibody. Once a white blood cell, known as a lymphocyte, has started producing its particular antibody, it can no longer divide. So if it can't divide, we're left with this finite number. That means a fixed number of white blood cells that are able to make each type of antibody. What we want to do is we want to create a cell that can both divide and produce antibodies. And we know that cancer cells are very effective multipliers. So let's have a look at this diagram. So we've got basically a vaccine occurring here. We've got the antigen, so that's the dead or weakened pathogen, going into this mouse or rat. And then as a result of it, we've got some white blood cells that are able to make antibodies. So antibody forming cells, that's just here. And then we've taken our tumour cell, okay? The tumour cell doesn't have the nucleus in it. We fuse those two together, the antibody forming cell and the tumour cancerous cell. And from that, we get a hybridoma. Don't confuse that with the monoclonal antibody. An antibody isn't a cell, it's something that's made by a cell. So when we fuse the antibody forming cells with the tumour cells, we get a hybridoma. Think of it as like a hybrid car or a hybrid bike. Okay, it's got features of both. It's a hybridoma. Once we've got that hybridoma, we then have clonal expansion. Okay, we're able to get lots of those hybridomas. All of these cells that, because they're multiplying because of the features of the tumour cell and they're producing the antibodies, the features of the antibody forming cell, we've got lots and lots of hybridomas. And then they're making lots of these monoclonal antibodies. So let's have a look at that explained in words. So the mouse has been injected with the pathogen, the antigen, those little markers on the pathogen that represent what it is, to stimulate an immune response. Okay, so it's working as a vaccine to stimulate the white blood cells to start making the right antibodies. The mouse white blood cells then respond by making those antibodies. The mouse blood is extracted and the white blood cells separated by pouring over beads and extracting the correct white blood cells. Don't worry yourself too much about that. The white blood cell is then fused, so the white blood cell that makes the antibody we're looking for is fused with that tumour cell, the cancer cell, in a petri dish or test tube. Okay, and the nucleus from the cancer cell has been removed. It creates this hybridoma, a cell that will multiply and produce the desired antibodies that we're looking for. Clonal expansion is the mass production of lots of those monoclonal antibodies, and those antibodies can be used in passive immunity. We talked about passive immunity with the antivenin, the snake bite. We can't make the antibodies that are needed, so we get the horse to make them and then they're injected into us. But these monoclonal antibodies could be used the same way in passive immunity. Let's go back to that picture. So we've got the vaccine with the antigen going into the mouse. It creates the white blood cells that can make the specific antibody for that antigen. That antibody forming cell, that white blood cell, the lymphocyte, is then fused with the tumour cell that's had the nucleus removed to form this hybridoma. Hybridoma is a key word. We then get lots of those cells, and we get all the monoclonal antibodies that could be used for that passive immunity. Other uses of monoclonal antibodies. So monoclonal antibodies can be used in pregnancy test kits. They're used to detect a hormone called HCG, which is present in the urine of pregnant women. Basically, the pregnancy test has basically got like a giant piece of filter paper in it. Okay, and some of the HCG that's in the urine attaches to some mobile monoclonal antibodies. They're mobile, that means they can move. When the HCG attaches to those monoclonal antibodies, they move up through the filter paper. When they reach that results window, they attach onto some more monoclonal antibodies that are immobile, that attach to the HCG. That stops the monoclonal antibody that was at the bottom from moving any further. It's attached with a blue dye. It creates a blue line in the box. There's also some other monoclonal antibodies. They just attach to urine. Some urine attaches to the monoclonal antibody, which is a mobile one at the bottom of the filter paper. That moves up to the control window. At the control window, there are some more immobile monoclonal antibodies that just attach to urine. That stops them in their tracks, and the blue dye that's attached to the original monoclonal antibody is shown in that control window. That shows that the test worked. And then the previous one shows that the person is pregnant because they have that hormone HCG. So it's all about mobile and immobile monoclonal antibodies attached with the blue dye that either attach to the hormone HCG or attach to just normal urine. Monoclonal antibodies can also be used to locate blood clots as they bind to the clot. So they can have markers on them, they bind to the clot, and then you can scan the person and find out where that clot is. Other uses of monoclonal antibodies, they can be used in labs to measure levels of hormones and other chemicals in the blood or to detect pathogens. Monoclonal antibodies can be used to diagnose and also treat cancers. They bind to the cancerous cells and help the person's immune system to attack them. In P4 we learnt about radioactive isotopes, and radioactive isotopes can be attached to monoclonal antibodies and the monoclonal antibodies then attach to a cancerous growth, and then it focuses the isotope that's going to kill that cancerous growth in that one area rather than killing lots of cells, healthy cells and cancerous cells going through the body. They focus on the cancerous cells because the monoclonal antibody is attached to them, kill those cancer cells without harming the other body cells. It means that with less side effects, we can use less of the chemotherapy drugs. It just focuses on that one area. However, there are disadvantages to monoclonal antibodies. Currently, there are more side effects than expected, and therefore they are not widely used. But there is a future for monoclonal antibodies because there's lots and lots of benefits, lots of different uses of how they can be used. Hope you found that useful. Thank you.